Institute, just up the hill. He did his PhD in quantitative biology at Georgia Tech. His bachelor's is in biomedical research, which is a new degree for me, at the Universidad Nacional Autónoma Mexico. He has published on many different subjects, fitting with the sort of wide variety of research that happens at the SFI. He's published on the origin of multicellularity, so how things came to have multiple cells, machine learning in biology. Sorry, my handwriting here is a little cramped. The division of labor in biological entities, and also biology and spatial arrangement or spatial structure, particularly topological structure, which is also new to me. And I should also mention that Pedro has longstanding interest in science communication and in public science. And so we'll get a little demonstration of that today. Join me in welcoming Pedro.
Thank you. Today I want to talk about very different topics, but all grounded in biology. As a biologist myself, I think these are very interesting topics, of course, but I think they also have relevance to other fields, especially in the natural sciences and maybe even in philosophy. The problem of biology, the central problem, is to study things that we call alive. So if you have a picture like this one or a painting, and then you try to identify which of these entities are alive, probably it's going to be pretty easy because we know about animals and plants, and we even have names for them. But then in this painting, there's also some things that are not alive, molecules, gases, etc. And the distinction between them might not be as obvious if we want to do a scientific inquiry into this division. So let's say if we abstract these classes of systems into two groups. Let's say one of them, we say that is a class of living things, and the other on the right is some class of non-living things. Then we can proceed with this and say, okay, then let's find what are like the general principles that are common across all of these entities that we think are alive, and also all the common principles or features of these things that are not alive. This has been, as naive as it sounds, but this has been one of the main attempts to characterize life. And at the end, what we desire from this approach is to find a kind of a list of properties, let's say reproduction, homeostasis, evolution, etc., that is different from the properties of the non-living things. And seems like a simple enough approach that it could work, but it has not worked for centuries now. No such list exists, because if you make the list too general, then almost everything is alive. If you make it too restrictive, almost nothing is alive. So there's no proper balance, really. So what could be another approach? One paper that I found super interesting that takes a different perspective on this is Morowitz's and Smith's between history and physics. They published this in '82, and basically what they were saying is that biology is some of the phenomenology that sits between purely physical processes and historical ones. And by purely physical, we mean the things that physicists call natural laws of the universe, or properties of, let's say, electrons or atoms that are almost independent to where these atoms are. For instance, right now I had just a sip of water, and when the water was in the bottle, it was not alive, I'll say. But once it's part of my physiology, it's part of a bigger system, and that bigger system is alive. But nothing happens to the water. The water is still the same, H2O at the molecular level. If you measure every single property of that water, it's still the same inside a living system or outside. So that already should hint at this approach that we might not have to care as much about the physical structure of something, but more about how it actually works. On the other extreme, we have history. History was a very big topic, but history in this paper was thought about as the phenomena that happens when there's some contingency at some point, and then everything downstream that point is restricted in its possibilities. For instance, I don't know, the invention of cell phones, the invention of the internet, allowed other things to be invented later on, but they were contingent on the internet and cell phones and even electricity, right? So these two, and there's no natural law to explain that. There's no universal law of the invention of the internet or the invention of electricity. We don't know if these things could happen or having found in other planets, let's say if there was life somewhere else. But we do know that, at least in theory, and so far we know, every single electron and atom will behave the same regardless of the planet that it is in. So biology sits here in the middle. There's many law-like phenomena in biology, but also there's many contingencies. Even the fact that we have 5 fingers and not 12 is a contingency on the ancestors that had first these limbs that we call hands. And we see that very clearly in evolution. Evolution really is a historical science. Even when we use mathematics and other more formal tools to study evolution, it's almost always a chronicle of what has happened. And the predictions that we can have in evolution are very limited. In fact, we saw that very clearly with the pandemic. In just a span of 4 years, so many scientists across the world tried to come up with formal models to predict the next wave, the next variant, and there was no such thing as a good prediction, really. Locally, sometimes we get success, but in general, evolution is a historical science. There was something also between this divide in physics and biology that a chemist, very famous Ilya Prigogine, pointed out, and other people had before him, which is that the ways physicists describe the world, the natural world, with dynamical laws, have time almost as a geometric parameter, in the sense that it is not really doing much. It is just the space where stuff happens. But in itself, time is almost a-dimensional. In a sense, we have an event, like a something that happens, let's say an initial condition, like the universe, the Big Bang was some initial condition of many things that happened after. And then we have another thing and another thing. But there's not really a clear causal connection between these. In the models, I'm not saying that in nature, like this is obviously not an ontological claim. So then you can have different initial conditions. For instance, these 3 lines in this panel A go forward in time because I just decided to make the arrow go that way. Well, this is Prigogine's diagram. And then I can have also the same initial conditions but go backwards. And there's a perfect symmetry, at least in the ways we study the systems. So this is important because the formal methods that we use to study dynamical systems don't say much about why something happened before something else. It just says stuff happens, and just by convention or by convenience, you decide that one direction is the future and the other is the past. But there's no natural way, or at least in the model itself, there's no way to tell which direction is which one. A quote I like that makes this very clear is that a motion unrelated to time, dynamics being this theoretical framework, a motion unrelated to time, or more strangely, a motion which proceeds in an intemporal time, which is kind of an interesting concept, a notion as paradoxical as that of change without change. So in essence, what it says is that once you define a dynamical law, you already have access to the whole future and to the whole past of that system you're trying to model. And in a sense, time doesn't mean much, really. What is interesting from the standpoint of the practice of science today, and since I would say at least the 16th century, 1600s, is that this is the way that we are trying always to model change in this kind of in-temporal time framework. So in a sense, dynamical systems are surprisingly static. Some of the first people that seriously tried to say something opposite to this idea were biologists, and we call them now, after many, many years, we call them, some of them vitalists, or which is kind of almost a pejorative way of saying that they were trying to invoke unnatural forces into a natural system. But really what they're trying to say when they, many of them, when they invoke notions like teleology, direction, desire, is not something magical. Maybe they didn't have the technical chops to make a formal mathematical model of this, but they were looking at the phenomenology, and many times that's where you start, and then you develop theories. And what they really want to get rid of is this complete symmetry between the past and the future, because what they observed is that an embryo always develops from the zygote into the fully developed organism, and not the other way around, right? And aging proceeds in a more or less regular way in almost every single species. And therefore, there's always a kind of asymmetry in time, but there's no theory, or there was no theory back then, that could account for this. In fact, Prigogine tried to come up with some of these theories. He called this the thermodynamics of irreversible processes, but in a sense, he didn't go far enough, but he planted a seed, at least to me, very important conceptual seed. So they really implicitly are contending with this symmetry, and to me that's what I take a lot of value of that position. In contemporary biology, I'm going to tell you, my colleagues, my biologist colleagues, they sometimes are even afraid of making claims that sound teleological or that have anything to do with purpose or desire, because traditionally, we are still in this realm, and in this realm, there's no desire. In fact, this complete symmetry tells you that there's no preferential direction. So, yeah, this was really an attempt to make not just a chronicle of something, comma, something as before, but something entails something later in the future, and this succession of events is not just random, it has causal effects in the world. And this issue is even deeper than just the phenomenology of biology. I will say it touches upon the mere foundations of how we do science today, which is that ultimately the more quantitative things are becoming in science. My PhD was in quantitative biology, but part of what I wanted to do is to say that it is insufficient to just make mathematical models. And part of the reason is that if you have a natural system, let's say this blue box here, and it has some internal workings that we're going to call causal implications. We don't know them yet, or we might not even know them ever, but almost the whole edifice of modern science is to find a formal system, and by formal, I don't necessarily mean just mathematics. It could be even a philosophical argument. It's still a kind of formal system in my mind that has some inferential implications. So in philosophy versus math, there's different ways of supporting an argument, but there are, right? So let's call those inferential implications. So the whole trick of doing science, in natural science, in many cases, is to go from the natural system to the formal one, And you do that by encoding some features of the natural system into the formal system. And once you have the formal system, then you study it, poke it around, change some variables, whatever, and then find behaviors that you might have not observed already in the natural system, or even the ones that you've already observed, and just see if they correspond to each other. So it is always an approximation, because this encoding, even though science and even mathematics seems to be a collection of very precise techniques and measurements, really the encoding part is more like a craft than an actual science or a technique. A craft because I could decide to model pandemics, for instance, with differential equations. I can use other tools, even like video games I could use if I didn't know anything about how to model differential equations. You can use many specific systems, formal systems, to represent the same natural system. And they might or might not be compatible. That's another thing. So let's take an example. You observe in some ecosystem that there's a population of rabbits and a population of wolves, and then you just measure how many of them are there across years, right, time, and you observe this oscillation. Then you can think of trying to describe this in more formal terms, let's say in mathematical ones, by just coming up with some equation. This is a very well-known equation. I'm not going to get into any mathematical details in this talk, but just showing you one such possible model. And this model, the output of this model is predictions of what's going to happen in future times. Now, as we already said, this kind of specific model, which is a differential equation, is just symmetrical in time, right? But still a formal system, and it's still very useful. In fact, I would say a lot of the progress in theoretical biology is still thanks to the use of differential equations. But then you can also think of, and let me make a remark here before moving on. I want to say that we can primarily understand nature through models. So this might sound strange if you're not studying mathematical biology or something, but I even think about mental models of, let's say, a society. A democracy is a kind of model of governance, for instance, and people have that kind of model in their minds. They have a model of the people that they're friends with, and that's why if they start to act strange, then it's like something's not checking with my model, right? So that's the more broad sense that I mean when I say model. Okay, so now let's study a little bit more specific examples of the kind of phenomenology we can observe in models. Because then if one of the primary roles of science is to understand nature through models, then for sure models should be important. So let's come up with a very, very simple model. I have a universe of, very boring universe with three entities. One is a cloud, one is a heart, and one is a star. These are just shapes. And in my universe, just to give it some structure, I'm going to allow some partitions of these entities. So a possible partition, for instance, is this one, where each element is just independent to each other. They live in their own little domain. And I'm going to call that system S1, system 1. Then system 2, I have a different partition. And you can already think about how to fill out all the possible partitions. Now, in this case, I'm going to say that if the heart is connected to the cloud, it also means that the cloud is connected to the heart. So there's a kind of symmetry here. And also, if the heart is connected to the cloud and the star is connected to the heart, then it implies that the cloud is also connected to the star. Nothing crazy, it's just a very simple description of these systems. Well, if the system is so simple, is it possible for it to behave in strange ways? Well, let's see. We have a scientist, and the scientist asks whether the cloud and the star are connected. If they observe the system 1, the answer is no, because they're not connected, as you can see in the yellow shade area. And then you do the same for all the other systems, and you get these different answers, right? It sounds pretty simple, because it is. Now let's say we take two of such systems, S1 and S2, and we ask the same question. The answer is still no. But what happens if we join them? This is just a symbol to join them. What is the resultant system? And by joining, you can literally just superimpose on top of each other. Just the yellow areas will be the sum of all the yellow areas of the independent systems. And then you end up with this system, which is the join of the two, but it's still very similar to the one we already have, because there's nothing else going on, okay? If you ask the question, are the cloud and the star connected? The answer is no for the independent ones, and also no for the joined one. So there's no surprises. You can try the same with these two systems, S3 and S2. Ask whether these are connected or not. The answer is clearly no. But then in the join system, the answer is yes, they are connected now. So might be not as crazy, but it is a surprise because there's a property of the subsystems that is not preserved once you make this operation of join. Now sounds very, it sounds very innocent of a claim, but it is at the heart of why a system can or cannot be composed and decomposed. Is again a kind of asymmetry now instead of time that before we were discussing on dynamics, is an asymmetry of composition of systems. So many properties that clearly exist at some subsystem level, once you have a bigger system, these properties are not preserved, many of them. There are some that there are, but here I wanted to give the example where they are not preserved. Now let's get a little bit more technical. We already answered this. We may call this generative effects. Generative because, again, we didn't put it into the model. It just kind of appeared. We were just observers, kind of external observers. Now let's do this with slightly more formal notation. I'm not going to go into mathematical details, but this is one way that in which we can represent functions, mathematical functions. And something that you can do with mathematical functions is that if you see in this function f that takes as an input x and gives you y, and some other function that instead of having as output y, it has y as an input, then you can just chain them together. So then you have this composed function. And you can do this in whichever way you want, as long as there's this compatibility of input-output pairs. For instance, here I just decided to represent this as a network. And I could say, well, I can also connect this subsystem to a bigger system, right? Some other arbitrary functions that are not very relevant right now. And then the whole system, I'm just going to call it a whole system, right? Now, what can we ask about individual and global functions in this case? For instance, what is the global role of a specific function, in this case, for instance, f, right, in the whole system? Well, as we said, we start with chains. If the outputs and inputs coincide, we just chain them together. We might call everything on the left of the function of interest sources and everything on the right targets. So if you take that graph that I drew before, then all the source targets chains are these ones. So I call them just functional chains around F. However, once you start to represent this in a network, because you just map every single possible matching, then you obtain a network. If you wanted to ask what are the sources and targets of F, instead of having this very simple one-to-one mapping as before, X goes to Y, the answer becomes much more trickier because upstream of X that I'm pointing out here exists this whole subsystem, and downstream of Y, this other whole subsystem. So this object here is not a function anymore. not in the traditional mathematical sense. Again, I'm not going to go into the details, but you already see that it's not an unambiguous X to Y mapping. Here, in fact, you could go from A to Z, B to C, X to C, and many other combinations. So then if you were to ask what is the function of F within the whole system, the answer is complicated. So it's not as simple as before. When you look at it locally, the function of F is to just take X and make a Y. Now it's like, well, it could make A into Z, or B into C, or X into Y, or even subsets like many to many. And those maps, in mathematics, the many to many maps are not functions. And even more, this is already one of those generative effects that we mentioned with the earlier, simpler case. This already is generative because the arbitrary composition of functions doesn't give you like a function in itself, right? It gives you something else. In this case, it's called a relation. Now let's do a little trick here also. Let's invert this function g. It goes from y to z. Now let's do the opposite. It goes from z to y. And now something else happens. Like what was before clearly a source, like a subsystem source and a subsystem target, is now in this brown highlighted subsystem is neither a source or a target. It's something in the middle. It's kind of a cyclic behavior here, because you can think of y going to c and to z and then back into y as having an output that then becomes its own input. So again, this adds more ambiguity. And if you keep playing this game, this is a very simple system, just 6 nodes and almost arbitrary arrows drawn. But it already behaves very different from what the individual building blocks were. So the second remark I wanted to make today is that a system of functions does not, in general, behave as a function. Now, why is this kind of detour into kind of mathy stuff relevant to biology and to the real world? Well, one, because we care about functions in biological systems. In the case, is very clear, for instance, in the case of biomedicine. If you have some gene that produces a protein that is faulty, and then the phenotype produces some disease, then in some sense you're saying, okay, the function of that gene when it was not faulty was to preserve that, the homeostasis or the stability of the system, but now that it's faulty, it has a different function that happens to be bad, right? It happens to be a disease in this case. Also, it matters because in evolutionary timescales, these mappings start to change. Just as we did this, we inverted this G. Evolution sometimes has mutations and other phenomena that change the structure itself of the system in very simple, minimal ways, but then the global effect in the whole system might be profound. Many mutations in many animals and in humans are just single point mutations, but they change the whole behavior of the system. Okay, an example is P53. It's a gene that early on when it was studied, people thought that it was the key to prevent many cancers because when it is working properly, it takes care of many processes that are important to keep cells alive and stable. And people in molecular biology and biomedicine, and I suffered a lot with these papers because you make these diagrams and they're very incomprehensible really if you try to get at what is the actual function of P53. Like, do I start here and then go through it and then go around and around and around and then go through here? Do I start here and then I end up somehow there? Like, what is the function of P53? Well, that's one of the main reasons I went into this previous detour of functions and compositions of functions, because now it might be that there's not a well-defined function of P53. Really, if you change its relationship with the other components of the system, you might end up with another disease instead of a cure, right? Because many people would thought that, well, if you kind of repair the faulty gene, then in this way, then there's no disease or no side effects. But no, in some sense, this detour that we just took before also points out at the fact that natural systems in general will have side effects that we cannot really control. Like when we inverted the G, we couldn't just have predicted that the whole system would change. Instead of sources targets, you have something else. In fact, that's something else that was labeled here as a relay, that we call relay, is something that I don't know has a name, if it has a name in the literature, in biomedicine, for instance. Another example is the same protein represented in a different way. For instance, P53 is now here. Before, it was more like a diagram that you show in an undergrad class, I guess, in biomedicine. Now it's a diagram that you might show in a, I don't know, statistical mechanics conference to show the topology of the disease network or whatever you want to call this. So the nodes lose a lot of details of the little bombs and colors that you have here, and they only preserve the relationships. But again, and this is just a subset of all the interconnections. I think these are around 25 or 30 genes, but the decision of where do you stop the description of the system is also arbitrary. Like we have 16,000 genes more or less. If you kept at the edges of this diagram, let's say here at V1, and then drew the connections of all the connections of this gene, then we might have much bigger system, and you do the same game with everything else, and all of a sudden you have almost like a percolation, which means simply that the number of nodes involved explodes. So an early discovery in biomedicine, and well, early, not so early, I would say like 30 years ago, 20 years ago, that people started to note is that when you have a system where you remove a single gene, let's say in E. coli or in yeast, they started to classify this knockout is the kind of experiment, the name of the experiment. If you do this knockout of the gene and assess if the E. coli or yeast survives or not, then you classify the genes as essential or non-essential. That was a very big, I would say, wave of experiments 20 years ago or a bit more. And I guess it looks kind of natural to do that experiment, right? If you remove some part of a system and the system doesn't work anymore, then maybe that part was very essential. So it makes sense. But then people started, as the technology evolved also, to be able to knock out two genes, tuples, then triplets, and so on and so forth. And it turns out that even at the double knockout experiment stage, you have this percolation of essentiality, and almost every single gene is essential in many systems, let alone if you knock out 3 genes at once, 4, 5. And these are 3, 4, 5 out of thousands, right? What it really tells you is that these side effects and these global functions that we were talking about are really integrated in most living systems. Living systems are not as modular and as independent in their parts as machines. If you have a car and something breaks in the car, you go to the mechanic, and very likely the mechanic will have a solution because it was this part that you need to replace, or it was that component that you have to replace. So in a sense, when your car breaks down, it's like a natural knockout experiment, and it's very easy to bring the car back to life. But in a living, and not only that, you can break many parts at once in the car. You can all basically total the car and still kind of rebuild it. It might be more expensive, but in principle, you can do it. But in a living system, you cannot do that. So again, the system as a whole doesn't behave as simply as we think individual components would behave. I'm just going to briefly mention some recent work I've been doing in a way to represent in general these kinds of interactions. Let's say you have components of a system, like the circles, and the squares are just transformations, which are the kind of functions that we were talking about. This was just for ease of drawing diagrams. For instance, if you have a bunch of circles and squares, then you can have a system, okay? Now, as we saw, the composition of the system, meaning the system as a whole, doesn't behave nicely as a regular function, as an input-output unambiguous line. So that's what we then wanted to study, the dynamics of the system. What are the behaviors that are possible? We think that two of the main behaviors that we have across all living systems are, one, cyclicity, that you can think about this as the way in which living systems recycle many of their components. If cells never recycled matter and energy, it would be a very, very expensive endeavor to just be alive. And if you also consider that many things, even though they're being converted into other things, let's say in biochemistry, some of these components are conserved. For instance, when I have an enzyme that transforms a substrate into a product, the enzyme itself is not consumed in a sense. It stays there. In fact, it can catalyze the reaction many, many, many more times. And that's one of the key aspects of the economy of the cell. Many components of the cell have to be used again and again. So once you have these two very basic features that we explain in detail here, but I just wanted to give you a taste of things that are preserved and things that are recycled, then we find what we call an organizer. And the organizer, what it does, what is the global function of an organizer? Well, here is where math stops, and then we just start the craft of doing science. And we just call two kinds of behaviors, one autosynthesis and the other autoanalysis. Autosynthesis means that you produce the components that you need, which all cells do, except, well, many plants that take energy from the sun. They heavily depend on the sun, and that's an external input. We also require food and energy and all that, but internally, like the instructions of how to be a human, how to repair a tissue, how to replicate DNA inside a cell, is self-contained. And we show that what you need is this synthesis, but you also need analysis, which is breaking down the components. If you had a system that only produces and produces and produces things, it will eventually explode. It will deplete all of the resources around the environment. And if you only had a system that only had autoanalysis, then you'll have a self-consumption process. And both of these things actually happen to the extreme in many biological instances. For instance, in cancer, there's a proliferation of cells that is uncontrolled. They just keep dividing. So this side of the diagram, the autosynthesis, is dominating the other. There's not enough cell death. Cell death is very important for all organisms except single cell organisms, because then the whole organism dies. But even there, it exists. Because sometimes we think that we just need to repair tissues and to produce more and to maintain it, but we also need to consume things. In fact, if you were to trace every single cell and every single atom even of your body today, and then just trace it with some crazy experiment, and you come back in, I don't know, 5 years, all the atoms, all the components will be dispersed across this planet. Probably some radiation outside of this planet will also take some of your particles. But you'll still be you. So something that is not tied to the material substrate is preserved. And that's really the central point that I think is also a big departure from traditional physics, which is that in physics, we started and we keep caring a lot about matter and what matter is. And in fact, a lot of times we think that the structure of a system determines its function, in biology even today. But here what we're kind of saying is that no, I mean, you almost can forget about the specific building blocks, but something else is preserved, a kind of description of this natural system in this mapping that we have of the natural and the formal system that is non-material. So in a sense, a lot of people criticize the vitalists because they were saying the same things but offering no kind of formal proof or no mathematics, and it's like, well, what you're saying is just almost that there's some magical force that makes living systems what they are. But really what we're saying is that there's a very natural way to think about this phenomena at both micro and system levels. For instance, in this simple system that I drew here, I want to obtain something more interesting because people will say, well, this is just a drawing, like a little child could have drawn it, and what are the interesting behaviors of it? In fact, if you start from some arbitrary initial state of the system, which I represent here on top, these ones and zeros are basically variables. In this case, just how many of these components are there at any time. And the Greek letters are just these functions, these kind of transformations of the whole system now. If I start from this one single initial condition and let it run through all possible functions that we define, what you see is that the tree starts to branch, and many branches go out and then come back, they converge. And this doesn't happen in traditional dynamics. In traditional physics, you would start with one set of initial conditions, your dynamical law, and you follow one single trajectory. So again, going back to the earlier discussion, there's a symmetry in time in physics, in traditional physics. There's no history because there's no contingency. And here you can see already kind of some seeds of contingency. I'm not saying this is history, obviously, but contingency is an important element. For instance, if I wanted to reach this endpoint here, I could take many alternative paths. I could go here, and then here, then here, and then there. Or I could go first here, and then here, and then here, and then here. And this is just a very simple system. It's basically this one here with 2 kinds of components and 4 kinds of functions. Imagine a system like E. coli with thousands of genes, thousands of interactions with its environment. Imagine human being with billions of individual cells, many tissues, cognition, many social interactions. What does that look like? Like, how do we describe that kind of system? Well, I would say at least not in the simplistic, ahistorical, non-contingent way. That could be a start. Now, this same system, besides behaving like these contingency paths, it can also behave like traditional physics. You start here, and if you take this path, then you can say, okay, that's just one trajectory. So in a sense, even though it's a toy model, it's not representing a specific system out there in nature, it can already kind of give us the hint that living systems behave both as historical entities and processes, but they could also have behaviors that are traditional and that they just go over time like traditional physics. So this is kind of the link that I wanted to make between the earlier paper I mentioned of biology being between physics and history, and also say that if you keep this network growing, many times you find yourself in a loop, like you come back to the same place, and that's also a central feature of living systems. Here, these red dots, once we let the system keep going, are the same exact state, so you kind of find an orbit. So in a sense, this simple system gives you linear chains, it gives you contingencies that go out and merge, and gives you cyclicity. Just want to mention a couple things about other theories of living systems, because there are many. I'm just going to mention 2, because there's some of the ones that are more known in the biology literature and also to me. One is autopoiesis. This was proposed by a group of Chilean scientists, and it was a model that was not mathematical, it was more conceptual, but I still think it's very important, and it had a big impact, not just in biology, but in other fields. Even I've seen books on the autopoiesis of architecture and the autopoiesis of the mind, et cetera, et cetera. The other one is a theory called MR systems that used a kind of math in which instead of you caring about the individual components, you focus almost exclusively on the functions, on these mappings that we defined back then, like F goes X, it takes X, makes it Y. Focus on the F, G, etc. functions. And so these two theories are also interesting because one of them was very conceptual and it resonated with a lot of people and their work. The other was very, very abstract. In fact, even mathematicians sometimes are intimidated by this kind of math called category theory. So if mathematicians were kind of intimidated by this subject, why would biologists care, right? So this one to the right stays on the periphery of the mainstream biology. So I think that what we're proposing is a kind of intermediate. We're not giving up completely mathematics. So in our paper, we describe formally what the system is and how it behaves, but also wanted to bring it back to the conceptual aspects, like what makes a living system alive. It's not its components, it's its behavior. In Mexico, we have this phrase of, I'm gonna attempt a translation, but well, in Spanish, if anyone knows Spanish, is dime con quién andas y te diré quién eres, which basically is like, tell me who your friends are and I'll tell you who you are. In a sense, it is focusing on the relationships that you have, not on the kind of individual, almost particle features that you have, right? It's like, well, tell me, what are your relationships? I also know that in English there's this phrase, birds of a feather flock together. That's kind of the opposite. And they focus on the substance. If the birds share this kind of feather, like if the structure is there, then they will behave in that way, right? So one goes, it's kind of an inversion of focusing on the substance and then the behavior later, or vice versa. And I think we need both perspectives. Like, of course, we would eventually like a specific treatment of a gene or a pharmaceutical compound that helps to cure some medical diseases and other things, but we also want to not tie ourselves to the specific gene or the specific molecule that is used, but understand it more globally, especially in a population as diverse as the human population. Many treatments that are effective in Asia might not be effective here, not because the gene is not there, but because the relationships that the gene has with the bigger system, the bigger physiology, and even cultural things, like how in some cultures, if you're a man and you go to the doctor, it seems like you're weak. Like all of these relationships to medicine in itself are important, but again, these other relationships is not just I give you the pill and then problem solved. I want to say something more about this modeling relation a bit more formally. You don't need to understand this again. It was basically the same diagram, but I want to show a distinction between a model and a simulation, because not only because simulation theory and all that is like in the mouths of everyone these days, but also the way we do science sometimes forgets about how do we actually make a good model. In a simulation, let's say you have the natural system on the left, S1. It has this function, this global function, let's call it, that takes X into Y. Then you make this encoding as a scientist. You take some encoding alpha of this Y, alpha of Y, and of this X, alpha of X. So you have the result and the input and the output, okay? But then to go from the input to the output on this right-hand side, you could choose an infinite number of functions, arbitrary functions. I could choose a G. And what is a kind of an example? For instance, in bacteria or in many other systems, but in bacteria, there are these patterns that we call Turing patterns that are kind of patches of domains of spatial domains of system A and B. It's almost like the spots on a cow. But you can also have the same phenomenology in oyster clams, in zebras, in many other systems. So in a sense, you're choosing an arbitrary mechanism because the mechanism in a zebra is very different than the one in E. coli. So this G is different. But you still get the same output. You still get a Turing pattern, right? That's subtle, but it's very essential because then what they call the congruence condition is that once you have this encoding method alpha that takes this f of x, and it is also here, then you're including something that is almost external to the system you're trying to model. Like it's not a correspondence directly to the model that to the system that you're trying to model. So that's a simulation. An actual model will need to account for the actual mechanisms if they do exist across these two different systems. And we also have many of these kinds of models. In fact, in biomedicine, we use all the time what people say are like mice models or yeast models of disease. You have the yeast, and if it has the same kind of gene regulatory network that the human has, then you can just, mess with that network and expect that the one in humans will behave similarly because they actually share both the genes and the structure. So that encoding is not just an encoding that gives you the correct output for the wrong reasons, like a simulation, but an actual model that takes the essence of the behavior of the model you were interested in and then gives you, again, the correct answer. So in this condition here, what you see is that alpha is doing all the work. There's nothing else. And in here we have alpha, which is the encoding, again, what you do as a scientist, but this G that is almost external. So that's kind of my third conclusion, that we ought to find models, not simulations of reality. As final remarks, just kind of remind you that biology sits between physics and history. Science is mostly an endeavor that deals with models, even if they're not mathematical, and that biology has this asymmetry in time from the past to the future. Thank you.